So it's either the port bio box or nothing.
I could try to deposit it away from the coral, and I do have not a lot of material compared to previous scoops.
So we have both. There's material in both port bio boxes, and I'm not sure we'd be able to get material into the starboard bio box.
We did it.
So, yeah.
Yeah, it's easy. Just flip your wrist around. Drop right in.
Okay.
Just as you come around, rotate at the same time.
So yeah, we could.
Try it.
All right. So they want starboard outer. Is that what I'm hearing? Or?
Just talking to Jeff, he says he has been able to get it in the starboard box before, and we're certainly willing to give it a try. But if that's your preference. Okay. You want to try it, Levi?
Sure. Yeah.
Are you ready?
Yeah. Oh, okay. Dan, they're going to leave it here.
Okay.
Understood.
You don't want to go for the starboard box then? Okay.
Okay.
All right. Copy. Closing up.
No,
That's
all right.
fine. You want me to wing inboard a bit?
Yeah.
So unfortunately, we've been informed that we really should not be combining the sediment and
That's good.
the specimen we collected, and even though they're from the same spot, and the reason for that being that
You're not
the
lined up.
Yeah.
Yep.
biological sample may have associates, meaning other organisms that live on the stalks and within the tentacles, etc. And so if we were to collect the sediment sample and put it in the same bio box, there may be organisms living within the sediment that would then be confused as being associates of the coral. And so that would kind of mess up the science. So we don't want to do that for certain. There's only one remaining bio box available, so that we have another, and because
secure.
we have another hour of the dive, we may see another biological sample we need to collect. So in the event that we don't collect one more bio sample, then we will take a sediment sample at the end of the dive. And we're going through a watch lead, so we're going to be here for a second. And so an exchange, what did I say? I'm sorry. We're going through an exchange of the front row. The pilots and nav and co-pilot, they're all switching. So in the meantime, maybe Cheryl and I will give some introductions to the back row. I'm Leslie Sauter from the College of Charleston in the geology department, and I'm the science geology lead.
And I'm Cheryl Morrison from the U.S. Geological Survey in Leetown, West Virginia, and I am the biology lead. And we are at the Stetson Mesa South site, and we're at 710 meters depth. And we are at the top of a large mound, and we've been looking at all sorts of different corals that grow on and around this mound. The mound seems to be formed most likely by scleractinian corals, which have been building structures over time, and then sediments get trapped within those structures and more coral grows on top, and eventually you get these mound-like structures. They're called bioherms.
The name bioherm just seems
All right.
too insignificant.
Pilot just to transfer some
yeah,
information
it's
to
true.
and just pass to me.
These are biological
Due to the
volcanoes.
current, what we're trying to do is keep the delta Z up
but
around 20. And
it's very
keep my
interesting
camera
because
angle around
they had never
70.
been
That way
explored,
we can see all
and
the tether in
there
front of
are
us, so it doesn't
hundreds of
sweeping
these
underneath
features on the
Syria.
Stetson
So if
Mesa.
we do hit a heave, we don't
One of our
come down and
colleagues,
smack
who
it.
was that? Chip Collier?
Yeah,
who
and you
said
also want to be worried
that
about
they were
heaving and
nicknamed
wrapping
Million
the tether
Mounds,
around
and
the
I
top
see
of
why
Sirius.
when you
Exactly.
look
So
at
if we keep it at
bathymetry
a
of the
delta 20
area.
and keep
We
you
were
in
hoping to be able
camera
to get
angle of
a
around
shot
70,
of our
Why is my
bathymetry
Z so
or map,
high?
but haven't been able
I
to
believe he used
post
it to push
that
into
on the
ground
other
a little
camera
bit,
yet.
the old station.
Okay.
Oh, the Z-app
And we were
bias
expecting
this
that this
morning,
would be
when I picked it
mostly
up, it was
Lophelia
31,
pertusa, which would be
and
the
I was still
colonial
floating up. That
stony
was because the
coral
current
making
was
these mounds.
Picking
kinda
it
But
up.
we've
pushing me up,
found
yeah,
that
so it would've
there's
changed.
also a
That
fair
was
amount of
significantly higher than
Analopsamia
that.
profunda. That's quite a different species as far as how related those two are, but they have very similar, they create very similar habitats and structure.
So if we ship loses DP, you're going to be turning to your starboard, and I'll just push out.
Copy that. Yep. Sounds good to me. And in order to keep you in the 70-degree view, we're probably going to have the ship moving.
We're coming
decent
up on
amount.
an
I
area
believe it's
with
stationary
more coral
currently.
now.
Yeah.
It's stationary, so I can put a move in. Let's
Looks
see,
like
330. just within 20
Analopsamia
meters.
right below us.
That works for me if that works for pilot.
Great. And then also, pilots, if we want to introduce ourselves, pretty good time. We could just let Washids know, and they can get ready for us.
And it looks like
Copy
our
that.
front row of our
Sounds like we
ROV
heard.
team is ready to give us their introductions. We'd like to hear from you.
All right, I'll start. This is Josh Carlson in the pilot seat piloting the D2 ROV, and my co-pilot.
This is Lars Murphy and our navigator.
Sean Kennison?
Over on the other side on video, Bob Knott.
And in the back row, I'm Emily Narrow on video clipping, and we are all with the Global Foundation for Ocean Exploration.
Thank you,
This is the
everybody.
bridge over.
We have a very talented team here.
Absolutely.
Fun to work with. Especially at 11:30 p.m. when
Did you
everybody's
say
a little
330
punchy.
degrees?
There's a lot of work that we do after the ROV
Okay, we'll
comes
move
back to the
20
surface.
meters
So
at 330 degrees with a speed of decimal 2.
process samples. We tried to summarize the dive that just happened. We planned the dive for the next day.
The video team is madly going through all the video and getting frame grabs of all the incredible fauna that we've seen,
As
the
well as
structure and the landscape.
and the samples that we take.
Yep. And our sample manager
RV
has
nav
to
bridge, move initiated.
figure it all out and keep detailed and careful records for the database. We photograph everything. We
Yeah, once you get settled over there, I
things,
believe there's
we
a sample
run-
in the port,
prepare
outer
for
and inner.
DNA
Are there any
samples.
samples in the starboard bio boxes?
Our
Yes,
remaining,
port outer and inner are full, and
Yes.
then starboard inner.
yeah,
So
We have
only
just
starboard
one bio
outer
box.
is the one left over.
Copy that. And I noticed that all the Niskin bottles haven't been fired yet. Are we doing water
Only-
samples?
Only
or-
if they request it.
Copy that.
Let's do we'll do a snap of this rat tail here.
Another Nizumia, rat tail.
Okay, come on up.
and earlier we saw a shark. It may have been the same one that we saw twice, or we saw two sharks. And Christian Jones has reviewed the video footage and believes it is Squalus cubensis.
Okay, I'm going to come up a slight bit
Thanks,
on my
Christian.
camera until I get you into view, and then I'll move it back
You
to
can,
70.
You can stay where you are.
Right.
Just back up.
Copy that.
This is watch one.
Go ahead.
Could we take a closer look at the coral that's in the lower left there? It's a little yellowish. Yeah, kind of centered up right now. Thanks.
Just come on in video.
Trying to see whether this might be. Okay, that looks like Lophelia. And up at the top of this mound, it looked like there could be some Madrepora. We have another Duva Florida, some more hydroids. A little bitty stylaster there on the left. Some sponges, oh great. And another Anthomastus or Pseudo-Anthomastus. That's the reddish coral in the center there, and right to the top, you have a bamboo coral on the left. Oh, goodness gracious, the more you look, the more you see. And several different sponges. Looks like some more of the bamboo coral that we've been seeing here and there. So there's this more stout bamboo, and then there's the more spindly bamboo.
ROV nav bridge move complete.
We have some orange cup corals in the background. And a fair amount of this bamboo coral growing all over here.
Still pretty spread out there, Pilot. Would you feel comfortable with another 20 meters or so?
You've
Yeah,
barely
you
started the
start
move.
But yeah, probably have another 2 minutes to swing.
Copy that.
I think that's
Even with this
Lophelia, yeah.
current?
Yeah, I mean, the current is just causing the layback. So, I mean, we can keep moving. It doesn't affect me.
Yeah, I can put another 20 meters. That'll probably put you around where D2 is.
Yep, that's fine.
And we have about an hour left on the seafloor.
Let's come out.
Bridge. Sure. Okay, 20 meters, 330 degrees.
So if
speed
you
decimal
have
two.
not been watching throughout the dive, you are seeing, what you're looking at is the bright white corals or living corals, and there are many, many different species that are living amongst the skeletons of dead coral, which makes up the framework of all the elevated material
Oh, look
you
at that
see in
sponge.
your view. That's about the largest sponge we've seen today.
Yeah.
Quite sure it
ROV
is.
nav bridge move initiated.
Some nice glass sponges in the background. It looks like this one is sort of commanding everybody's attention.
Anybody have an idea about what sponge this might be?
Let's do a quick zoom here.
Lots of little crinoids on the sponge. It's got quite a bulbous looking exterior.
Well, that's different.
Yeah. And then a Duva Florida on the top. A soft coral. So Scott France says it looks like a demosponge, Heteroscleromorpha. Scleromorpha. And wow, 4 beautiful glass sponges right behind it. What a beautiful scene. That looks like a hydroid.
Copy that.
And another lace coral. And a soft coral. Ooh, now that one looks like a primnoid there. Pilot, can we hold right there a minute?
Sure.
Thanks
I
a lot.
agree with you, Cheryl. That looks like a Caligorgia, perhaps.
Just come in tight.
Caligorgia
I feel like
or
lasers off.
oopsie. Plumarella, yeah, that's it. That's the one. I think that's the Atlantic, more common version of something that looks like Caligorgia. Thanks, Steve. Gosh, we've seen a lot of diversity today. This is great.
ROV nav bridge move complete.
It's coming out.
I just spoke with Nav, and they said we've covered about 450 meters. I think we probably hit the 400 mark during somebody's lunch. So sorry we didn't report that earlier.
Or change of watch.
Change of watch or lunch. So
Another cutthroat eel passing by.
we have about a little under an hour left. And we've traveled across, we started at the top of a small mound and then moved toward a second mound that was slightly higher in relief. Climbed up and over that, and in between mounds we saw low depressions that had virtually no coral communities, just sediment-covered coral rubble. And then we climbed a very steep face of the third mound that is the highest of the three. So it's been sort of a increasing mound experience, and different types of coral structures here, and certainly the coral communities seem to have varied. Certainly comparing this community to the one on the lower or the deeper community on the shortest of the three mounds, the first one.
I'd say that there's been more diversity as we've reached the top, and then we've gone from a Lophelia or an Elipsamia dominated colonial coral to Lophelia, and it looks like we have a Bathypathes in the center of the, or a little bit to the right now. Bathypathes black coral, and then that yellow coral up in front might be something we haven't seen yet today.
Okay, come on in, video.
And we're seeing a lot more structure here than we've seen on any of the other mounds. So the corals were on low-lying piles of coral rubble, and here these are much larger dead coral skeletal frameworks that have much larger communities surrounding and on top of them. They're higher up in the current.
So we saw Bathypathes, the species black coral we were just looking at there, the big feather-looking red coral. We saw, it might be a different species, but the same genus, quite a bit deeper, about twice as deep on Sunday.
All right, go ahead, Ed.
And This octocoral looks different than anything that I've seen.
Right, go
Maybe
ahead
a
and video.
Paramaricia. Got a little Nuzumia rat tail swimming there in the background.
Yeah, let's zoom here video.
It's a pretty one.
Very pretty. Also looks like the polyps are coming out either side very flat, like that Swiftia that we looked at before. Yeah, if you look closely, there's lots of tiny brittle stars. Very light pink color. Goodness, I would have
They
seen
look
those
like the
without-
polyps.
Yeah.
They have good camouflage. We've seen a lot of very tiny brittle stars today. We didn't notice that in the deeper dives previously. We often found one large brittle star dominating a living coral.
Okay, let's come on out.
All right, pilots, Stereo should be settling. Are you ready for another move?
So this is
Ready
Lophelia.
when you are.
Yep,
Great.
looks like
Away.
Lophelia, and
Nice
then
specimen of
Copy
it.
that.
possibly Bathopsamia, that orange coral, cup coral
is
just
bridge
underneath
over.
the Lophelia.
Yeah. Okay, 20 meters at 330 degrees, decimal 29.
Thanks very much, Nav. If we can just kind of continue to take a look at the whole top of this ridge for our remaining time, that would be wonderful. So we've reached our last waypoint, but we are now at the top of the largest one of the mounds that we plan to explore today. So we will just use the rest of the time to run back and forth over this and get a good idea of all the corals that are here. It looks like we have quite a diversity, so I think we have plenty to look at.
What did you want to see, Watchley?
ROV nav bridge move initiated.
Yeah, so we're still climbing a little bit. We're now at 715 meters.
Do we want to try and follow this feature up or continue northwest?
Yeah. Yeah, if we can follow this feature, that would be great.
They want to go more east.
Copy that. We can cancel the move and reline Sirius and move that way.
Sure.
Does that work?
Sure.
Great.
You want me to spin around? Are you looking good there, pilot, at 40?
Something around here.
Is that a
if you
call
wanna
to the
turn your
bridge,
heading.
over?
Alright, I'm going to start coming to starboard.
Coming to an easy stop.
And yeah, couple that. You can just center in the direction we want to go, and then I'll put in another move.
Copy that now. Should we bump Sirius's sonar out a little bit and
There
see
are
if there's
mini
anything
mounds on the bigger
further
mound.
ahead, or are we
Yeah.
happy with 30?
I can do that. Stand by one.
More of a question for
ROV nav
discussion
bridge,
rather
we
than...
are stopped. Over.
Now it's worth a shot.
Gives you a sense of how the rubble constructs the mounds, and then the sediment infills, and eventually it becomes solidified.
I'm sitting at 045.
Oh yeah, our first sea star of the day.
Yay.
Great eye, Bob.
Yeah, Sirius doesn't pick up.
I wonder if
Whole
Chris
lot, but
Ma
it
is
looks
still with
like-
us.
Or even maybe
I don't think this
05,
is very high, so
Yeah.
we can just.
It's not supposed to get any higher either.
Yeah.
I would just head this heading.
Copy.
Something along those lines. All right, come on in.
Great.
This heading as far as yours, pilot?
Yes.
070?
Yes.
All right, swinging around at 070.
It looks like a bat star.
Cookie cutter.
Cookie cutter. Yeah, that's a great name.
All right, I'll put move in 20 meters at 070.
Copy that.
Saw some of these on the Charleston bump. They weren't yellow, but they were genus. I'm looking this up at Peltaster. I don't know if anyone's out there who can confirm that that's a Peltaster. Very similar.
Okay, we will move 20 meters at 070 with a speed of decimal 2.
Oops, looks like a slice of lemon.
Little slice of lemon.
A lemon cookie.
Yeah,
Try to
lemon
come
cookie.
in tight here, Bob.
Aw.
So there's been some discussion in the chat room about which species of octocorals
Are we now bridge,
have
move,
a lot of
initiated.
little brittle starfish, little ophiuroids that live on them. And
Come full wide.
looks like acanthogorgids often have lots of little brittle stars, but the plexurid, which is what we saw earlier, might not be as common a thing. Hello.
Hey, this is Chris Ma.
All right.
Hello, Chris, how are you?
Good. Well, as people have said before, if you shine a starfish up in the sky, I will come running.
We were trying that last week, but we couldn't
bat
quite
star.
reach you. You were too far away.
Oh, I was asleep, most likely. Yeah, you guys are close. That's indeed a cookie star. That one is in the genus Plinthaster. So,
Plinthaster,
since
okay.
there's only one species in the Atlantic, Plinthaster dentatus, that is certainly what it is. Plinthaster, the name, actually, plinth is Latin for stone, and those are smooth. The abaxonal plates on those animals are essentially like flat, smooth plates. I mean, they're literally like flat, smooth, like rocks. So that's probably where that name originates from. And that's a widespread species in the Atlantic. They vary quite a bit, but they occur on both sides, both in the sort of tropical Atlantic, but also on the European side. Honestly, I don't think we know a lot about them. So, I mean, in the sense of what they eat or whatnot, it's probably not terribly dramatic. It's not. They're probably, they probably feed on epizoics, maybe some microalgae or some colonial animal. But observing them would obviously be, really something interesting for such a well-known species. But this is one that's known and varies quite a bit across. There are lots of actual species of different cookie or pentagonal-shaped goniasterids, and they all seem to look alike. So Ceramaster, Peltaster, even Posinaster parvis has a really distinctive pentagonal shape, and and this has a very sort of pleasing aesthetic to it. But they also all have very sort of distinctive characters. They just vary quite a lot, and so some of them, such as Peltaster and Ceramaster, look a lot alike, and so they can. But the differences tend to be in terms of covering like granules or whether they're smooth or what have you. And this becomes more complicated in the Pacific, where you have, I think, maybe another 4 or 5 different genera of cookie stars, if that's what you want to call them. A good spotting, and I think that one's probably a classic tropical Atlantic species. So, it's one of the many things that I come to expect in the Atlantic from this area, and looking forward to it. I was just looking through through your earlier expedition photos and saw some quite interesting asteroid species. And as I was telling the participants in one of your prior expeditions, these expeditions to the Gulf of Mexico are surprising to me because I had thought that based on the collections here at the Smithsonian and other places that we had surveyed everything, that we knew everything. And in fact, like I said, even from just this week, there are images of animals that I don't recognize.
Wow.
That's not because I'm not familiar with them. It's because they're unknown.
Wow,
And
that's amazing.
There's something you guys saw on a sponge that I was like, what is that? And so, I'm hoping you'll see one again, and that we'll be able to collect it or at least get better data on it. So, but even getting observations, if we saw Synthaster dentatus hunched over a coral or some feeding event, that would also be very happy and add to our knowledge of these sea stars.
Huh.
Surprisingly, I think we actually have more data on the Pacific ones than we do on the Atlantic, and the Atlantic ones are better studied in a sort of specimen-based way.
Wow, that's interesting.
Chris, sorry.
I was just going to say, you would think that we would know most of these species out here right outside our back door, but it's true that there are so many that we still have a lot to learn.
Chris, I was wondering...
I think it's always... Right.
I was wondering if the yellow of that sea star is diagnostic for the species, or do they vary in color?
They vary in color. I've seen them white. The yellow is very common, and I've certainly seen... Yeah, it's a very commonly encountered color for that species, but, they're solid-colored. And in the Atlantic, they can be orange. So it depends, I think. But yeah, like I said, the area, these kinds of areas are very surprising to me because, there's perhaps an illusion of that we know what's out here, and in fact, there are always new things to be discovered, and even I forget that, and and I've seen quite a lot of these from collections, and so sometimes submersibles are the best way to go because you see things that you wouldn't ordinarily see, and
yeah, you see behaviors and you see colors and habitats and
associations.
You are hitting it right on the head. Yeah.
Yep.
That's exactly right.
Yeah, it's amazing.
And I don't speak only for sea stars, but everything from sea spiders to, to every other kind of animal that you have out here, or even protists, for that matter.
Looks like this is the end
there
of it.
are all sorts of things that, like that swimming brittle star you saw the other day.
Yeah.
Turns out it has been reported before, but never in such a beautiful visualization.
Yeah, that was amazing, wasn't it?
Yeah.
Oh, I saw that, and I as it turns out, I happened to be I was at the Echinoderm conference while I was in Japan, and I had to send it. I showed it to 4 of the world's experts.
Kind of turn around.
They were just right there, and they were just
and head back the way
their mouths were just dropped open. And fortunately, they were able to tell me which paper to go to because I assumed it was a novel observation, and it has been seen before, but not since the 1980s, and. And even then they said they weren't sure based on the video which one it was. So there could easily have been, that could easily have been a new species, or, it's certainly not something commonly known. And the only place that has been published, that paper was published in 1990.
Wow.
So that's from observations in the 80s. So it's like some of my favorite TV shows. They haven't been seen in a long time. So that's the thing is you seeing stuff like that is just gold. From a marine biology and marine biodiversity perspective, it could be. It seems almost sort of natural history, but it might have some broader significance. It was reported, for example, that sideroid sea urchins fed on stocked crinoids, and that might seem like more of a natural history note, something more localized, but it turns out that that could actually have been the motivation for why crinoids, the feather stars can swim. So that could have been a large part of
maybe if we took
their
like
adaptive radiation, or could have had a huge impact on their evolution. So even something that appears to be relatively localized as an
Come over the
observation,
top of
might
that
have wide-standing,
slope.
Broad
Just so we
application
wouldn't be seeing
later
the same
on. So
terrain,
basically, even though it seems
we'd be
novel,
heading
I mean,
right
like
towards the
not
top
just
of
not,
what we believe
but
is
something
the
that seems
local
very, just kind of neat, might actually be
Yeah,
very,
any direction that
have very
totally
broad
doing no problem.
implications later on. That's what's so great
for you,
about
pilot,
marine biology.
Want
We're
to go
seeing
330?
things that
Yeah, that kind of cut back
that
across
seem kind of
slightly
like,
ahead
oh,
of
that's kind of interesting.
waypoint
And then
4.
the next thing you know, boom, it becomes
We
a
pretty
critical
much
part of a
came
huge
almost
hypothesis.
directly
Yeah.
east from waypoint 4.
It's just really interesting how this topography is becoming so rugged now. We
Center up.
Yeah,
seeing
so I'll
more
swing
sort of
back to
flat-lying
330 or around there.
landscapes with
Or stop wherever you feel happy.
colonies
and then we
popping
can
up,
just
but
go
now
that
we're
way
seeing
next door.
major
Swinging
colonies,
back,
and the
pilot.
topography
Coming to the port.
is just up and down and up and down.
these
We're looking at
huge
a
mounds.
gully, and at the base of the gully was a pile of rubble of the old coral pieces.
Well, thanks so much for calling in, Chris. I hope that we're going to see more sea stars, asteroids over the next couple weeks. And
yeah, I'm looking forward to it. There's a sea star that I described from this area called that was a predator on corals, and I've only ever seen the specimens. I've never actually seen it alive, so I'm looking forward to hopefully that you'll encounter it.
I hope so.
We'll see. Have a good day and have a good expedition. I think you guys are doing a great job, and I'll catch you guys a little later.
Okay, thanks very much, Chris.
Thanks, Chris.
You have a good day. Bye. Bye.
Bye-bye. That's Chris
back
Moss
uphill
from the
this way.
Smithsonian Institution. As you can tell, he's a sea star expert.
This is bridge over.
Just back from an echinoderm meeting in Japan.
And for those who are not biologists, an echinoderm is Echinodermata is the phylum
Okay, we're going to move
to which the sea stars
at a
belong,
bearing of
along with their
330
cousins, the
degrees, that's
urchins,
speed
and the
the
sea cucumbers, and the
sea
brittle
cucumbers.
stars, and the crinoids.
The crinoids.
And we've seen all of them at some point in our expedition.
And I wonder if Chris has had a chance to see the sea cucumber outhouse that we got to witness on Sunday.
In action.
Yeah, that was pretty exciting.
All right, 500
that,
meters into the dive.
ROV nav bridge
Out of
move
an
to
initial 650 we had proposed, but we had to cut down about 100 meters of that due to the currents and where we were able to land at the start of the dive. So we're nearing our goal of what we thought from the bathymetry was the shallowest point here. Seems like we're still climbing.
70,
but we're kind of
sitting
wandering
happy.
over this
Feeling
broad
comfortable?
top of
Very
the tallest of the three mounds that we have crossed.
Are you
And
looking
it's,
upstream right now?
it's
or
just
uphill?
amazing how vast this top area is of this mound.
Very dynamic.
Looks like sonar might even pick.
You really
something
feel
up
like it is
west.
growing upward with all of
We
these
might want to
coral
head a little more
communities,
towards like
much
315,
taller than what we
330
started with,
after this next move is
just reaching
done.
up into that
Up, yeah.
current.
You can see all the particulates in the water column, and
Do you want
that's
me to
what
follow
the
you around
corals
to-
will be
Yeah,
feeding
you
on.
can follow me around here.
Alright.
So this is definitely upslope, but it's so this is the west watch lead, which is definitely upslope, but
That looks good if we're able to head that way, west or northwest.
it's just away from the patch of all that coral.
I
So
imagine we'll run into more if we
Yeah, we
want to keep
just
going
want to
this
keep
way.
going up.
Right.
Thanks.
I suppose if you want, I can cancel the move, and then we can just move west.
how much more do we have in the move? Just
It was only 20 meters, right?
yeah, you
It
can
was
just
a 30-meter move,
Oh,
so
30.
Yeah.
we're like 12 meters in.
yeah, it's up to you.
Just let it ride.
I mean, this is a pretty large feature if high pack is
Yeah.
correct.
You just
Yeah.
let it ride.
A little hot here. Yeah. What speed was that move at? 0.2?
Yeah, decimal 2.
Okay. Go ahead. Mm-hmm. Which box is open?
Starboard outer.
Starboard outer.
Okay.
We
we can make an attempt at that. 15 minutes should be enough time. Did you copy that, Nav?
Oh, yeah.
You're slightly out of my
Yeah,
screen,
I'm
by the way,
backing
pilot.
up.
I saw your lake pool, so we have an idea of what you are, but
So here's another large area of mostly dead coral matrix with some octocorals and sponges and a cutthroat eel swimming by. This has been a very typical scene as we've been moving, especially on this largest of the mounds. We've been here on different parts of this mound for the last couple hours, I'd say. We have some glass sponges on the left and some more bamboo corals in the center now. That looks like another big glass sponge coming up. And maybe a more dead bamboo coral in the background. Ooh, and there's another beautiful
lovely
sponge. Oh, and then in the background there's a white sponge with zoanthids.
And what is the red thing down at the base of the sponge? Can we get a quick zoom on that? Is that
Come
just
on in video.
I wonder if that's a hagfish. I've seen a hagfish that's that color. I don't know why it would be here though. Or any other ideas of what that might be?
looks like an eel?
Yeah.
Trying to bring my head around, see if we can see more of it.
Nice look at the dead coral and how it's encrusted with the iron and manganese oxides that are in the water that precipitate and coat anything that isn't alive, basically. Even the sediments get coated after a while if they're in place for a long time.
Somebody's hiding.
Somebody is definitely hiding.
It's a cup coral there on the right.
It's a good hiding place, too.
Yeah.
Yeah, I think that's the best we're going to do here.
I think we're not going to see him. We didn't scare him enough.
Right, come on out.
Would you like me to put lasers
Thanks
on?
for that look, pilots.
Sure.
Coming on.
And we have a Liopathes black coral on the left. It's actually orange in color. And they get their name black corals because their skeleton underneath that tissue is actually black.
All right. Guessing they're pretty close to being
Yeah.
done. Let's do a quick zoom here.
And the white sponge in the center of your view now is one that has yellow zoanthids growing on it. And we've seen this many times now in the past couple of dives where you'll have a sponge that has these zoanthids growing on the outside.
They've been yellow here and they were white on the escarpment. It looked like beards on some of the sponges.
We have bamboo corals, which are the corals that are kind of in the view of the sponge.
Christmas lights.
And Ren Salgado said this is a heterochrone sponge, or I don't know if you how to exactly to pronounce that. heterochrone.
And perhaps a stalked crinoid waving behind the sponge. Scott Prance says. Not sure where he means, but we'll take a look.
And that was the yellow sponge. That was the heterochrone, not this one.
Yeah, that crinoid I saw would be here. It's just coming into view.
There it is.
Oh, yeah.
Yes.
Right behind the sponge. A stalked crinoid.
It's one of the few echinoderms that are often preserved in the rock record. The stems or the stalks of this particular type of crinoid is made of little poker chip plates. And each plate has a radial pattern with 5 points, pentagonal pattern to it, and each species has its own arrangement. So we can actually identify, at least to the genus level, the crinoid from which the stalk or the fossil comes, and they're much more resistant to erosion, and they don't break down. So they do get preserved in the sediments, whereas all the other features you're looking at rarely do get preserved. If you go to a museum, you'll see plenty of fossils of them, but those are the museum pieces. We don't see them very often.
Right, come on out.
We just see the little poker chips.
All right, should we make that move uphill?
Yeah. Copa, you want to start to rotate?
Yeah, we want to swing around to your direction there, pilot?
Yep.
Right, swing around to 220.
Oh, we can do something.
Yeah.
Looks like anywhere up here would be good.
Okay.
Hey, when you're ready, Sean.
Great. I'll put in 30 meters, 220.
Copy that.
Bridge over.
Let's do
we love
snap
moves.
here real quick.
Okay,
Let's
we
do a
will
snap
move
here.
30 meters at a bearing of 222 at a speed of decimal 2.
Let's do a snap zoom here.
That looks more like an Alapsamia to me.
All right, let's come out.
And Chuck Messing said that the stalk crinoid with 5 undivided arms is most likely Democrinus.
ROV nav bridge move
Thanks
initiated.
very much for that, Chuck. We are now at 718 meters.
Copy. What kind of shark did you guys see earlier? Do you know?
A squalidae shark
Oh.
is what we saw earlier. Squalus cubensis, I think somebody said.
I think that's correct, yes.
This is bridge over. No. Just to clarify, no issues with propulsion at this time. We just have a trouble alarm on our fire panel in the propulsion room. We are sending an investigator over.
So it's the fire alarm related.
Yeah.
We're changed a little bit. Would it be better for me to come to port now a couple degrees to line up?
Yeah, it looks like we've got some more little white style-starred lace corals coming up here.
If something happens.
If we lost DP, if you have time, take the turn out.
All right.
If
so
you
go
don't,
all the way to starboard.
If you don't, and they immediately lose DP, then just keep your, just whatever you can to get lined up.
Copy that.
And that I'll swing around to get in position.
Gotcha.
We're seeing some more cutthroat eels. We've seen many of these today. Sometimes in
video?
Concentrations of 7 to 10.
Yes.
Yep.
They likely live within these coral structures.
Maybe we can take a look at this big octocoral ahead.
All right, come on in, video.
Wow, what a beautiful coral.
Partial.
Looks like we have another one of the Plumarella or Caligorgia behind it. On the left.
It's another bamboo coral, I believe.
Oh, is it?
Mm-hmm.
Oh, yeah. I can see the bands now.
All right, everyone, we have about 30 minutes left on bottom.
This would
through
be
today's dive.
Tighter shot when you get more stable.
You can see
I'm
that
not
it's
going to get more
very
stable.
planar, so a little
You
bit
can
different from
you can
the one we
snap in if you want.
earlier.
That's about the best we
it's
can
growing
get.
in one plane like a fan.
Anything else would just be blurry.
But
There's a
possibly
ton of
or
current
likely
today
also in the D clade.
and a ton of stuff, so I can't really set down. So it's going to be difficult.
And next to it, for comparison, that really wispy thing, I believe,
This is Bridge over.
Platerices in the S1 clade bamboo.
Okay. So we've been seeing that
Yes,
off and on
the
today,
forces
haven't
are
we?
looking
The
very good. We have the
Platerices.
current as our strongest factor
Yeah,
directly
that's
on
I
the
think
bow.
what's been most
The
abundant
wind
today. All this really
is
wispy
very
stuff
light
that
and
you
also
see.
on the bow.
And one of the reasons it looks
And,
so wispy, first of all, it's got a very
all of the forces are on the props, which is, can handle it,
sort of
and
large
everything's
fan we're
looking really
looking at
good.
next to it, you'll see that the distance between individual branch points is really long, and so you get parts of the colony that go for a long time before they branch, and so it certainly makes for all these wispy tips.
Ah, yeah.
You are beginning to go off to my port. Do you want me to swing around to get you into view, or you
No,
plan on coming
I'm
back?
almost done here.
Copy that.
All right, come on out, video.
Maybe. It looks like more Lophelia behind that, the white coral that's creating this framework here.
ROV nav bridge, move complete.
Right at the bottom of the screen, we just saw another
Right,
pancake
but
urchin.
it's at 1945. If we don't
Now you
have
see it.
another sample, that's when we want to start.
Let's do a snap here real
Nice glass sponge right in the middle. And that looks like an Elipsamia, the other sclerotinine coral.
And a
to the left.
Sidereus. Another pencil urchin.
Another little round sponge in the background. And a lot more of this coral rubble that we've been seeing all day.
quick. Right, it's coming out.
At one point, we found flat-lying, rocky
so
type.
at this point we're
I say
kinda
type because
close-
I'm not positive
back
it's
close to
true rock,
1.4.
but areas of ledges
maybe we
that
wanna go
looked
more
like
west,
compressed,
possibly,
compacted
if
coral.
there's anything there to look at.
And we have
Yeah,
a
I'm
sample
coming
of that,
back
right?
around.
Yep, we got
So
a
we'll
sample.
be able to tell you a little bit more about that
You
tomorrow.
want to try like a 315
That was the only place we saw it,
300?
so I'm glad we came across it at
Let's
all.
see.
And that glass
Yeah,
sponge that we
as it
saw was
turns.
Africalistes. Thank you, Zach.
There's some more glass sponges.
You even do.
I mean, this.
245, 240.
Yeah, we can do that.
How's that look? So, yeah, Copa, you want to line up at like 240?
Copy that.
Let's go for that.
Coming to starboard.
And I will put a move in.
This bridge over. Yes.
Settling at 240.
Okay, I understand 30 meters at 240 degrees, speed decimal 2.
It's like a bamboo coral that's kind of laying down.
Maybe he'll start growing vertically. We saw that on the edges of the scarps on the Blake Escarpment. The bamboo coral would grow outward at first and then take a right angle turn and start growing upward.
Straight up, yep.
Yep.
We have a few more.
Small fish center, bright lasers.
Yeah, we've seen a bunch of these. We'll take a
Oh
snap
yeah,
here.
we have a small rat tail here on the soft sediment.
ROV nav bridge move complete, and then just to follow up with you, the alarm appears to have been a false alarm. There's no issues with the ship at this time. Over.
That sediment's a lot coarser than what we saw out on the Blake Escarpment and Blake Ridge, more on the Blake Ridge area. Much lower velocity currents out there so that the fine materials can settle out. And here, there's just too much velocity. The clay-sized particles don't have a chance to settle out. They're just whisked away with the current.
Chimera top left.
Yeah, go
Oh,
ahead.
yeah, we have another chimera. We saw 2 of these early in the dive, and now it's been a while. Again, this one's very small. We saw the ones we saw previously were also small. This looks more similar in color pattern to the first one that we saw. The second one was a little bit less differentiated in color.
You can kill lasers.
Lasers coming off.
I think the Chimera's having as much trouble with the current as I am.
Much more energetic at this depth than those that we see in the much deeper waters.
Well, there's a lot more current.
Yeah, he's trying really hard to hold his own in the current.
All right, come in on that, Roland.
Now he's getting pushed a bit.
Beautiful.
So these guys are known as ghost sharks. And they're also called ratfish, spookfish, or rabbitfish. But they're part of the Chimeriformes.
When you're good, Roland.
Good.
Yeah, it's coming out.
Now that was a great look at him.
Go ahead. It will be interesting. Let me see if I can find a spot where I'm not going to damage too many corals.
So Chimera's closest living relatives are sharks. But today, in our modern era, we don't really find them in shallow waters, only in deep waters. So it's always a treat to see a chimera.